Paclitaxel or Taxol is one of the most widely used drugs we have for breast cancer. It was discovered in the bark of the Pacific yew trees in the 60s, and over many decades was developed into one of our best cancer drugs. It's used because in a large fraction of patients, meaning as many as 50%, it can control or shrink tumors. However, it's not 100%. And so over the years, people have tried to find out why it's working in some patients and not others, and have been largely unsuccessful at doing that.
Cancer cells have to divide in order for the cancer to grow. Normally, they'll make an exact copy of each of their 46 chromosomes, which serve as the instruction manual for the cell, and then they'll divide those evenly on a molecular machine that's known as the mitotic spindle. The mitotic spindle is composed of microtubules, which are targeted by paclitaxel. For decades, it was thought that paclitaxel exerts its anti-cancer effects by freezing the microtubules of the mitotic spindle, which causes the cells to get stuck in mitosis and then die. But when we actually looked in the tumors from breast cancer patients that were treated with paclitaxel, what we found was that the concentration of drug that got into the tumor just wasn't high enough to freeze those microtubules and arrest the cells in mitosis. Instead, the cells would proceed through mitosis, but instead of dividing in 2 directions, they could divide in 3 or more directions, which would randomize the chromosomes and lead to cell death. Tumor cells commonly have accelerated genetic evolution, which means that their chromosome number and content tends to vary over time. And this makes it easier for them to adapt to different environments. However, we also know that there's a limit to the rate of change that cells can tolerate. And so their hasty evolutionary strategy also makes them vulnerable. They're susceptible to treatments that increase the rate of change to a lethal level. And about half of breast cancers have an inherent rate of change in their chromosome number. And our paper provided evidence that it's these inherently unstable breast cancers that are sensitive to paclitaxel treatment because it increases the rate of change over a maximally tolerated limit. And whether or not a given tumor is inherently unstable can be determined based on the patient's initial biopsy. And so this can potentially be used to help predict whether that specific patient will benefit from paclitaxel therapy.